Hello students, welcome to the class of Eduauraa. I am Sunil Kumar, your biology faculty for this session. If I draw this as the chromosome, each chromosome basically consists of, there is a centromere, and apart from centromere there are two sister chromatids. What I will be doing, I will be opening one of these sister chromatids. When I open it up, I get something like this. This is the structure. And after that I get something like this, and then we put it here. Let's put it in this way and here. So when you look at these structures, you can understand most of the structures very easily. Now in this diagram you can recollect many of the parts. This is nothing but it is the opening of the chromosome. When I open the chromosomes, I can see all these parts visible. You might be wondering what these parts are. So students, let's start with the very basic part. There is a sugar plus phosphate plus nitrogen base. Sugar, phosphate and nitrogen base, they will together make up something called as nucleotide. Number of nucleotides will line to each other, they will be joined to each other, and they will form a structure that we call it as a gene. And when the gene interdivides among each other, it forms a structure that is called as the DNA. This DNA is going to coil on a special protein that is called as the histone protein. And this histone protein is positively charged and the DNA is negatively charged. Because of this positive and negative charge, what happens? There is attraction between the two. And finally DNA coils further, and further it condenses to form something called as solenoid. It's like a telephone wire, so it's a solenoid fiber. This solenoid fiber will undergo further condensation to form a structure that is called as chromatin. And finally chromatin condenses to form chromatid. Two chromatids, they join to each other at a point called centromere. And finally what we get is the structure of chromosome.